Good morning, friends. A warm welcome to all for a very brief presentation on neurohumoral transmission, which is going to be presented by Dr. Videsh Pratap, who is working as Associate Professor at Gokaraju Rangaraju College of Pharmacy, located at Hyderabad. At the end of the session, the learners or the audience will be able to understand the definition of neurohumoral transmission, meaning of some important terms which they frequently encounter during the discussion of this concept, steps involved in neurohumoral transmission, types of neurotransmitters, and final inference or conclusion from this concept. First of all, we discuss the definition of neurohumoral transmission. So, this is nothing but the transmission of impulse through synapse and neuroeffector junction by the release of some chemical substances or humoral substances, which we call them as neurotransmitters. So in short, it is nothing but the transmission of impulses via synapse and neuroeffector junctions with the help of neurotransmitters. The diagram present in the slide depicts how the neurotransmitters help in the transmission of impulses across the synapse and neuroeffector junctions. So by this diagram, we can visualize how the impulses are transmitted through the gaps called synapse and neuroeffector junctions. Next, we discuss the meaning of some important terms which we come across during this discussion. First among them is neurotransmitter. Neurotransmitters are chemical messengers that help to transmit signals from a neuron to a target cell. Second one is synapse. It is nothing but the gap present between two neurons. The neuroeffector junction. It is another gap which is located in between the neuron and end organ. The next one is action potential, which is very important as far as this concept is concerned. This is nothing but change in membrane potential or electrical potential, which helps in the transfer of impulse across the membrane of a nerve cell. So this is called as action potential. So these are some important terms which we frequently encounter during this discussion. Next, the reflection spot. So, listeners, I want to ask one question to enlist the steps involved in neurohumoral transmission. Take a small break and list out the steps involved in neurohumoral transmission in your notes. Hope you might have written some important steps involved in neurohumoral transmission, which we will continue now. Basically, there are 6 steps which are involved in this process. Those are axonal conduction, neurotransmitter release, receptor event, initiation of post-junctional activity, destruction or dissipation of the transmitter, non-electrogenic functions. This is the pictorial representation of the mechanism of neurohumoral transmission. So in this picture, the neurotransmitter present is the acetylcholine and its targets are nicotinic receptors which are present on the post-junctional membrane. Now we discuss in detail about each of these 6 steps involved in neurohumoral transmission. First among them is the axonal conduction. It refers to passage of an impulse along the nerve fiber. So a suprathreshold impulse initiates a localized change in the permeability of the axonal membrane. So that localized change can produce either depolarization, which is nothing but influx of sodium ions from the extracellular surface to the intracellular surface, or repolarization, which is nothing but the efflux of potassium ions from the intracellular surface to the extracellular surface. Among these two changes, the depolarization actually leads to the action potential. So this action potential, once it's generated, gets self-propagated, and in that process it is going to conduct along the axonal fiber. The second step is the release of neurotransmitters. Once the action potential arrives the nerve terminals, it triggers some changes in the membrane of the nerve terminals. So those are influx of calcium ions. So that influx of calcium ions triggers or initiates the release of either excitatory or inhibitory neurotransmitters into the neuroeffector junction or synapse. So the action potential, once it reaches the nerve terminals, causes the synchronous at a time release of several hundred quanta of neurotransmitter. Depolarization of the axonal terminal triggers this process. The third step is the events which occur at the receptors located on the post-junctional membrane. The released neurotransmitters from the nerve terminals cross the synapse and bind with the receptors located on the postsynaptic nerve terminal. Then the receptor undergoes configurational changes, resulting in the opening of channels present in it or present in them. So this causes either the influx or efflux of ions. The fourth step is initiation of post-junctional activity. So the changes which occur in the post-junctional cell are of two types. So it can be either post-synaptic, excitatory post-synaptic potential, that is EPSP, or inhibitory post-synaptic potential, that is IPSP. EPSP produce action potential in postsynaptic nerve, whereas IPSP doesn't result in action potential. So, for the generation of action potential in post-junctional membrane, EPSP is very essential. The fifth step is destruction or dissipation of the transmitter. So once the action of the transmitter is completed, there is no need of the transmitter to present at that junctions. So immediately it gets either degraded or removed from the area or location. So sometimes some neurotransmitter gets locally degraded with the help of enzymes. Example, acetylcholine. Some neurotransmitters removed from that location by the uptake mechanism. Example include norepinephrine. The rate of termination of transmitter action governs the rate at which responses can be transmitted across a junction. The ultimate step is the non-electrogenic functions. So the continual quantal release of neurotransmitters in insufficient amounts to elicit a post-junctional response probably is important in the transjunctional control of neurotransmitter action, which we call them as non-electrogenic functions. So these are the 6 critical steps involved in the neurohumoral transmission. Next, we discuss about the classification of neurotransmitters. Based on the type of actions produced by the neurotransmitters, they are classified into three types, namely excitatory, inhibitory, and both excitatory and inhibitory neurotransmitters. The neurotransmitters which produces excitatory postsynaptic potential are usually termed as excitatory neurotransmitters. Examples include glutamate, aspartate, nitric oxide. And the second class is the neurotransmitters which produce inhibitory postsynaptic potential. They are called as inhibitory neurotransmitters. Examples include glycine, GABA, that is gamma aminobutyric acid, serotonin. Some neurotransmitters can produce both excitatory and inhibitory postsynaptic potential based on the location. They are called as both excitatory and inhibitory neurotransmitters. The good examples for this category include acetylcholine, norepinephrine. So the overall conclusion of this topic is the neurohumoral transmission actually helps in conducting the impulses from one nerve to another and also from nerve to the end organ or effector organ. So in this way, this process helps in transferring the impulses or messages from the central nervous system to the various peripheral or end organs and ultimately helps in functioning of the various visceral organs. So all the functions of the visceral organs majorly dependent upon the neurohumoral transmission. So maintenance of homeostasis is going to be helped by neurohumoral transmission. So this is in brief about our neurohumoral transmission concept. Thank you, my dear friends. Hope you all understood. Thank you. Thank you very much.